the Science Notes podcast on OAR FM Dunedin, brought to you by the Division of Sciences at the University of Otago. I'm Dave McMorrin. Over the next half hour, I'll be chatting with a graduate science student about their research, their passion for science, and the things that fuel their creativity. Well, hello there, and welcome again to Science Notes. My name's Dave McMorrin, and this time our guest is Jervee Punzalan. Hi, Jervee.
Hi, Dave. Kia ora.
Thank you for coming along on a surprisingly sunny Dunedin day in March.
Yeah.
It's a lovely day.
Thank you for inviting me.
You're very welcome. So Jervee is 2 years into a PhD in chemistry.
Yep, that is correct.
And we are going to be talking a bit about what she's been up to for her research.
Yep.
But to start off with, maybe we could talk a little bit about how you've ended up in Dunedin, because you're not from Dunedin originally, are you?
Yep. Okay, so I'm originally from the Philippines. So my passion for actually the country, New Zealand, started in 2018. So I attended a conference in Queenstown.
Oh, nice.
Yeah. It's the first conference. It's my first conference in New Zealand. And immediately I fell in love with the country.
Right. Because Queenstown, of course, is representative of all of New Zealand.
Yeah. It's really a nice city, and it's tranquil with a city vibe. You know, it's really a complete balance of everything. And so I was like, I'm looking for a university to do my PhD at that time. And so I was like, I wanted to do it here. If there is a university in Queenstown.
In Queenstown.
Well, I don't know anything.
No,
about
that would be lovely.
Yet. Yeah. And so I was like, there's no university. So fast forward, pandemic happens, and things and plans get postponed a bit. And then after that, I realized, let's just do it. And then, it's still pandemic, but sure, let's just start doing it. And I started looking for topics, of course, that interest me. And, this is also a funny part or interesting part, because my previous students, because I'm working as a lecturer way back in the Philippines as well.
Okay.
Yeah. So my former student is also studying here at the University of Otago and doing her, that time, master's in biochemistry.
Right.
Yeah. And now she already graduated as in PhD in biochemistry. And so I was like, how was it studying in Otago?
You have spies.
Exactly. And she was like, it's so fun and all. So I kind of went to the website and started looking for supervisors. And so it was the KCG group that kind of hit my interest.
Yeah.
And of course, I did some research about my supervisor, Keith. And I feel like it's very important also to have a good dynamics with your supervisor first. So before really diving into what you're going. And so, I did some questioning and kind of interview about the past students of Keith. And and I was like, let's just go.
Let's just go.
Yeah.
And so you came here in 2021?
2023.
Oh my goodness, 2023.
I know, it's like 5 years
in the making. It seems like you've been here for ages. But
it's 5 years in the making. I mean, starting from 2018.
Right,
So
yeah. Cool. So you're here, and the group that you're working in is kind of an analytical chemistry group as much as it's anything.
That's correct.
And it's using various different machines to try and analyze all sorts of things, really.
Yeah.
You're particularly interested in proteins.
Yep.
Why proteins?
Okay. I'll be honest with you. So before coming in here, it's not my original sort of research.
Right.
Yeah. So I'm supposed to do something related to AI and machine learning sort of things, and marine organisms and sort of things. But don't get me wrong. That's also under Keith. That's also under Keith. It's just that when I arrived, the project is about to end.
Right.
And so I have to I have to start with a new project.
Yep.
Which makes sense, right?
Yep.
Yeah. So I have nothing against protein because I'm already I'm already doing it. And I remember during my first year of progress reporting, Keith was like asking me, do you want to change your topic? And I was like, no, I'm going to fight for this. I already started this. I did a lot of research. So like, to be quite honest, it's like a whirlwind experience for me because it's really new.
Yeah.
I can say it's really new. Like, I really started from the scratch. To be honest. Like, yeah, especially the first year of doing my PhD. It's really kind of challenging for me because I really have to study about proteins and the instruments that I will use, and literally everything is really new because I wasn't really previously exposed to, for example, handling other instruments such as the Raman and the near infrared, and also using some software to analyze them. I really was not exposed to those. I was actually quite surprised that Keith accepted me as a PhD as a PhD student. I was like, oh, thank God somebody accepted me to do the PhD. Anyways, so why protein? So, well, let me just, sorry about the long introduction about that. But I think it's important to kind of have a perspective of what's I'm doing right now. So, proteins are, I think it's important part of our body. Mm-hmm. And it's, it's also present in all living organisms. It's the biomolecules that are made up of amino acid connected by your peptides. And I think all the important processes in our body are based on the synthesis of proteins, or at least the building blocks of proteins. Yep. And I feel like it's really important to study this sort of biomolecule. Mm-hmm. Yeah.
But in particular, you're interested in extracting the proteins out of, in one example, flaxseed. So linseed, flaxseed.
Yeah, that's correct. Yeah, flaxseed.
So why is there an interest in being able to extract proteins out of things like that? Out of
everything, or any other source? Something?
For what is the reason that you were doing it?
Okay, so we this is just statistics. Like, predictions to 2030 states that there will be a boost of demands for plant-based proteins.
Right. Yep.
So that is being fueled by environmental concerns, sustainability concerns, people being, of course, I think it also includes something about social-cultural aspects, population growth, and since I'm here in New Zealand, I think flaxseed has a huge potential to become a good or a nice plant-based protein source for human consumption. And and I think, aside from that, I think because flaxseed is also well known to have good effects to cardiovascular health, what else? Gut health, and also a good source of antioxidant.
Yep. I take flaxseed oil pills every morning.
See?
They're supposed to be good for me.
Wow. omega fatty acid. Omega 3, to be specific. Yeah.
And getting protein out of plants, as you say, because there's concerns about the sustainability of relying on red meat and stuff for that.
Yeah.
Another option, and we've had people on the show previously talk about this, is bugs.
Sorry?
Bugs. So insect protein.
Insect proteins.
But I think we've had people talking about that. We've also had people talking about the challenges of convincing the consumer to eat protein that's coming from crickets and what have you.
That's true.
Whereas from plants, it seems like an easier sell.
I can say it's yes and no.
Yeah.
Because, they said that like 50% of the reason why people are not really trying plant-based products is because of taste. So it's really one of the factor for that. So it has a lot of, I feel like there's a lot of things to do in this field, to be really able to improve, of course. So that's one part, like taste. It's I know it's not part of my study now, but I feel like in the future it will be. So I feel like I'm contributing to that better, good, or improving the quality of life, that sort of thing.
Yeah, so, saving the planet.
Why not?
Why not?
Yes.
One of the things that the notes you sent me talked about is the way that you're extracting the protein from things like the flaxseed is using something called pulsed electric fields.
Yep, that is correct.
So tell us about that.
Yeah, so pulsed electric field, it's a non-thermal technique that utilizes high voltage pulses to to give you because I know we have some non-science listeners, so it's like sapping the cells.
Yep.
Yeah. So you use electrodes to be the source of that high electric source, and then with this one, PEF has been used in the area of food science and food industry in terms of food processing. Well, it's known for its applications such as, for example, pasteurization and enhancing flavors in food. I think it's been used, or there's, I'm not particularly 100% sure, but in the US, they got some like commercial juices that are really PEF extracted. And since these are not using heat treatment, so they kind of preserve the flavor and some of the, let's say, nutrients coming from, let's say, fruits or vegetables or whatever it is that they're extracting.
So fundamentally what the process is trying to do is break open the cells and release the contents.
Yep, that could be also one of the effects of PEF.
And is that what you're trying to do in order to get your proteins out?
Yes and no.
Yeah.
So that could be also one. But for me, it's more of, since also one of the effect of PEF, because it's also increasing the conductivity of the samples and because it's underwater, it's also giving additional charges, or it's increasing the polarizability of the sample or whatever plant material it is, and then it's adding that effect of changing conformations of your, let's say, protein or secondary structure of your proteins due to differences in polarity.
Right.
Yeah, and so you have a different interactions happening. So like could be high increasing hydrogen bonding depending on what the interactions or whatever is present in there.
Right. So the proteins initially are kind of, say, stuck to other things.
Yeah.
And this process helps free them from the other things.
Yeah.
So you can separate them away.
Yep.
Right.
Yeah.
By changing the electronics, if you like, of the molecules.
Yeah. And also because the product or the sample that I'm currently handling is not pure. So it has also some interactions with carbohydrates.
Yeah. Yeah.
The other polyphenols.
The other biomolecules.
Exactly, the other biomolecules.
Yeah.
And we're trying to kind of deduce what could be the possible interactions that are happening in there from the different samples that we have extracted and synthesized. So, yeah.
This might be a question that you don't know the answer to, but if in the process of extracting them, you're changing the shapes and the electronic properties, does that have an effect on, because presumably the point at the end is to eat the proteins in something, does the changes that you make in that process, does that affect downstream the ability of the protein to do be nutritional the way you want it to, or to?
Oh yeah, I think that's an interesting question though. But I feel like, the next stage of this research is actually doing some sensory, and really incorporating them as a food. As food ingredients, like for example, for breads, for milk, yogurt, So since, I hope to partially answer that this year. So I will be having my last year this year. I'm doing some, I think I mentioned to you that I'm doing some food science experiments nowadays. And so I'm also looking into the functionality, properties of the products or the samples that I have. And I hope to relate them to the spectroscopic data that I already have. So
the spectroscopic data, these are another set of machines. These are ways to try and understand what those structures are of the proteins.
Yeah.
Because that would be really interesting, it seems to me, that if you have a particular way to extract proteins out of linseed that people are already doing, and that provides a product which tastes a certain way when you incorporate it into yogurt, and then compare it to yours.
Yep.
And whether the taste is the same. Because to a large extent, the way that things like proteins taste are quite related to their shape. And the way, because the shapes are what determines how they interact with your taste buds and stuff.
That is so true. In fact, so we had a recently published paper. From that paper, we kind of give like a rough what do you call this? An estimate of like what are the shapes or at least the secondary structures that are present or dominant at least.
Are they long and thin? Are they blobby or whatever?
Yeah, for a certain extraction, because we did different sort of extraction. We did different. So we use different electric energy input for our extract.
Sure. Yep.
So we kind of wanted to know, are those energy enough to, let's say, to produce a lot of alpha helix, beta sheet? Because I feel like those are important factors, especially when you're considering them to be part of food ingredient. Like, for example, this is just from literature, of course. I haven't tried doing this. I mean, but for example, if you want to incorporate them as bread ingredients, for example. So if the food ingredient is rich in, let's say, beta sheet, so you will expect that your bread to be crunchy and, firmer.
Right.
But if the food ingredient is rich in alpha helix, you will expect that your bread is kind of soft and mushy and something like this.
I didn't know that. Yeah.
Yeah. So I kind of wanted to know those sort of things as well. And, that is only for bread. So we're not yet talking about, yet say, milk or yogurt and other food that these ingredients could be incorporated or infused. So so that's why I think it's important for as you mentioned, the shapes and how it's going to affect the.
Yeah, I mean, so not only does the shape, because I mean, the what I know about proteins is more about how they interact with things like taste buds and stuff.
Yeah.
But the fact that whether the protein molecules are long and thin and helical, like an old telephone cord, or whether they're kind of more flat and sort of brick-like, affects the actual texture of the bread.
Yep.
That's really interesting. And obviously important, right?
Yep.
If that's the long-term goal of being able to use this technique to extract the proteins for things.
I totally agree with that. Yeah. That's I would want to be part of that project in the future. Yeah. But of course, I want to graduate.
You're going to finish sometime too.
Yeah. Because actually, that's the next stage of this research.
Yeah.
So my research is actually part of a bigger research. So the next part is doing the sensory and incorporating it into.
So these will be academics and research students in food science and in that department.
Yeah, that's their.
Who will do all the tasting and the blind
That's
test.
their part.
Yeah. Or
So
the trained tongues and noses and stuff that they have over there.
Yeah. And sometimes they also call for students. I've been part of that one time case where they ask for students to taste their, that was pasta, I guess. So, yeah.
So you're 2 years in. You've got another year to go.
Yep.
Hopefully.
Yes.
Collect up all the data. Write it all up. You've already had, 2 papers?
Yep. Yay.
2 papers is great. That's really good. So what is the plan after that for you?
Well, hopefully I got all the things that I needed to be when I go back, because I'm still affiliated to a university in the Philippines. So I'm affiliated at the University of the Philippines as actually an assistant professor, and then so I hope to carry all the knowledge that I gain. And I'm actually thinking already as early as now to propose a project and eventually be able to secure a grant in collaboration with hopefully the food and the Department of Science and Technology in our country.
Right.
Hopefully, I'm aiming for that as early as now, because I feel like that's the only way to go when you're under the academe. Because I also have to advise students when I go back.
Yeah.
Yeah.
Doing boring things like teaching and stuff, I suppose.
Exactly. Well, it's my passion, so I feel like it could be a bit boring, but for me, I think it's fun.
Yeah.
Experience. That's why I like demonstrating.
Yeah, I understand. But the plan is to continue somehow doing the research that you're doing now.
Yeah,
when you go back.
I enjoyed it.
Yeah.
I really enjoyed. I was not like, whew. I'm so thankful that I was able to. I don't know. I feel like everything happens for a reason, and that things are going for the good of whoever it is. So I was like, oh, it's good that I wasn't able to really work on that project where I initially supposed to work. So I was like, oh, this is better, so much better. I like it. I mean, more.
Good. Excellent. Well, thank you, Jervee, for talking to us about what you've been up to, and all the best for the remainder of the PhD and for whatever's next.
Oh, thank you.
And thank you, everyone out there, for listening. You've been listening to the Science Notes podcast, brought to you by the Division of Sciences at the University of Otago. To listen again or to find other episodes in this series, go to Otago Access Radio's website, oar.org.nz, or to your favourite podcast platform.